concepts that you've heard about, and in a particular context which may be easier for biologists than some of the very general mathematical ideas. So we're going to see questions about more than one locus being involved in changes, in evolutionary changes. We're going to see again examples of short-term and longer-term evolutionary change, reversal of evolutionary changes as well, I'll mention, and some of the concepts that I introduced in the previous lecture as well, like trade-offs, will come in. But first, I just want to make clear that in biology, we use the term sex, the word sex, to mean more than one thing. You've heard about asexual organisms, and I mentioned them too. And so we talk about the evolution of sex, meaning the evolution of reproducing sexually and not asexually. That's one meaning of the word sex. But here, when I say sex, a lot of people think we shouldn't use the word sex, we should use the word gender. Here, I mean male and female. So evolution of separate sexes means why do we evolve male and female individuals separate from one another? So forgive me if I don't say gender, because I never remember, but it's a completely different concept. These are, we're talking about an organism which is a hermaphrodite, sexually reproducing organism, evolving into a species with individuals that are male and female with separate sexes. And this is just to give you a concrete example. In a plant, I will again concentrate on plants because these kind of evolutionary transitions have occurred many times in plants and many times in animals too, but the most familiar animals to us, like ourselves and Drosophila, made this evolutionary change from whatever was their ancestor in this state to having separate sexes very long ago. They have a long ancestry of having separate sexes, and so we can't study the evolutionary change. But if you look at this phylogeny of the plant genus Silene, it's just one example. Others could be used, but my lab happened to work on this one. You can see that we have hermaphrodite species here, and some of the others are hermaphrodite outgroups, are mostly hermaphrodite species. And species, which I've labeled dioecious, crop up in a group here. So presumably in the ancestry of these 5 species, they evolved separate sexes at one point in time since the split from these species here. And interestingly, in Silene, it's a particularly interesting genus because it seems to have evolved independently into different sets of species in the genus. Some of the species that are not dioecious are also sexually polymorphic, and they're gynodioecious, the one I mentioned before. In case you don't remember, it means having females along with hermaphrodites. So here's a female, and here's a hermaphrodite. If you can see it all, maybe it's not clear enough, but the hermaphrodites have female parts here, and they have anthers producing pollen, the male function here. The females have the female structures, the pistils, and the anthers are little shriveled, non-functional anthers. This is a polymorphism within any population you look at of this particular species. And another word for the hermaphrodite is cosexual, meaning they have both sex functions. Whereas in the dioecious species, these ones, this is the male and female flowers. The male flowers have no female parts that you can notice except early in development where they stop developing soon. The female flowers, again, their male parts stop developing very early, and when you look at a flower, you only see the female part. That's Silene latifolia that my lab work on, and here are its sex chromosomes, which should look pretty familiar to you. We know that in humans, for example, we have an X and a Y chromosome. They look different from one another. In humans, the Y is a good deal smaller, about a third the size of the X. In plants, typically, and in this plant, the Y happens to be bigger than the X, but it looks different, whereas all the other chromosomes come, as you know, in pairs that look the same as one another. And you probably also know that the X and Y chromosomes do have one part where in meiosis they pair together towards the tip, both in humans and in this plant, and they have crossing over events, the recombination events Brian was talking about, but they're restricted to this part, which in humans is very small and includes just a handful of genes, whereas the rest of the X has hundreds of genes. In this plant, we don't know the numbers of genes, but we do know that they pair and cross over at the tip, but most of the chromosomes that look visible when you see them in meiosis don't do that. So I want to think about the question of how we get from hermaphrodite, which we symbolize like this. Is that symbol familiar to people?
Oh, it's the badge, but this is...
If we start with a population which is sexually monomorphic, co-sexual, and we come to a sexually polymorphic male and female situation, a dioecious population, what needs to happen? Sorry, you have to speak loud. No, it's not a change in gamete size. That's a good point, and I'm glad you mentioned it. The hermaphrodites have male gametes and female gametes. For example, here they have pollen, which is the male gametes, or contains the male gametes in plants, and ovules. That is all assumed to have evolved long ago. We're assuming that these are organisms that have male and female gametes back in their ancestry, as indeed is true for flowering plants or for animals. But we're just looking at the gender of the individual organisms, whether they produce both kinds, male and female gametes, or only one kind. So it's a very good point, but it's not what I'm trying to focus on here. We're assuming, yes, the developmental pathway's got to change, but in what sort of way? For instance, to make females, what have we got to do? Sorry? I'm not assuming any sex chromosomes. That's another very good point that I'm glad you raised. We're assuming a co-sexual, sexually monomorphic individual. So it would be individuals like this that produce both gamete types as developmental program of the flower in a plant, for example. There's no sex chromosomes. We want to try and understand separate sexes, and I'll come to sex chromosomes shortly, starting with an organism that doesn't have it. It shows how we are so familiar with separate sexes and sex chromosomes. We think of that as the ground state, but you've got to expand your mind and think of a ground state where none of that existed. We're just a co-sexual organism in the ancestry, which must be the ancestry of most things, many things. So to go from hermaphrodite to female, what have we lost? Sorry? We've lost the development of the male parts, right? So we could go with what we call a male sterility mutant. Mutation, and we've now got female. I hope I heard you right, what you're saying, right? So in thinking of the population genetics, which we'll come to in a bit more detail in a moment, what will happen? Will this mutation go to fixation? No, it won't go to fixation. Why? Yeah, you have to have males. If you're a female, you won't have any offspring if there aren't some males, or in this case, we will actually have a polymorphism established with hermaphrodites. The original co-sexes would not disappear because they would be the only source of male gametes. And as you heard earlier, and as you know, every offspring has a male parent and a female parent in a diploid organism like that. So you'll end up with a polymorphism. So we've achieved a sexually polymorphic population, but we haven't achieved what we're looking for, which is a contemporary dioecious population. So what has to happen to get all the way there? Somebody else? Well, no, I mean, this can be reproducing by cross-pollination, but where are we going to get the males from? We've got the females, so we could say in a dioecious descendant species, here are our females, they're okay, but where are we going to get the males from? These could revert back to hermaphrodite, that's correct. So we could draw that in, but that's just, that's going to take us back to there. It doesn't give us males to have the reverse of the male sterility mutation. Yeah, another mutation, yes. I'm not asking for the advantage, I'm just asking for what kind of mutation now. The advantage will come. Yes, a female sterility, thank you. So we have to have a female. I should have drawn the hermaphrodite on this side, really. We can say we have several of them. And then this one, we'll have to have a female suppressing mutation. In order to get males. And of course, you'll see immediately that we could have the opposite order. We could have a polymorphism with males and hermaphrodite and co-sexuals, and then we have this one mutate with a male sterility mutation to give us the males. Is everybody with me there? Because this is really critical to everything else. So what we've shown here is to reach a dioecious species like this, dioecious population, we didn't succeed with one mutation. We had to have two steps. We had to have first females and then males, or first males and then females. Two steps, or more than two, of course, because maybe this is a gradual reduction in several steps and this is a gradual reduction, right? It doesn't work with a single step. And that's why, one of the reasons why this is an interesting problem, it's not an advantageous mutation that we can think of, like being able to run faster to escape predators. It's something which involves two different changes before we arrive at a state which we know this transition has occurred repeatedly. It's not just a very, very rare, strange occurrence. So what we want now to make a genetic model is we want to understand why those sterility mutations will actually spread. You should be kind of surprised. And here's a quotation from one of the scientists who worked on this fairly long ago, Georges Valderon, French scientist. How can a gene or a set of genes which reduce the fertility of some individuals attain non-negligible frequencies? It should be quite surprising that these are very bad kind of mutations. I'm talking about, in this simple crude diagram, complete sterility through one sex or the other, when the co-sex had good fertility of both sexes. So that's what we're going to look at first. How can sterility mutations spread? So I'm going to start with how will females invade hermaphrodite populations, and in the problem class we'll consider males, but I'll mention this pathway, males invading co-sexual populations too. Now, we have to think of two possible, well, we have to think of the genetics. What might be the genetics of these mutations? And it's been known, and very well known for a long time, that in plants, at least, male sterility mutations that you find in crop species and genetically studied species are often mutations in the cytoplasmic genome. It turns out in the mitochondrial genome, you can have mutations that cause male sterility. Cytoplasmic male sterility factors, often called CMS factors. And these are extremely important in agriculture, where you want to make male sterile individuals of a co-sexual species because you want to breed hybrids and you don't want them to self-fertilize or have any of their own pollen on there. If they're male sterile, it enables you to put the pollen on from another strain that you want to make hybrids. That's how hybrid corn is bred, for example. And so I'm going to look first at that case for a little while, think through that case, and then I will come back to this, which is probably what's happening in the cases that of evolution of separate sexes. But just to show you that a complete sterility mutation can have an advantage. In fact, this is an extremely simple case in its simplest form. We can imagine that we have a hermaphrodite or a cosexual genotype of the mitochondrial genome. So it's a haploid genome, essentially, or you have multiple copies per cell, but it's, they're essentially all the same in the different cells of an individual. It's transmitted maternally like a haploid genome. And you can assign fertility of any number, so let's call it 1 for the cosexuals, and let's say females have a different female fertility. And let's assume that that's all that's going on. Why might females have a higher fertility, if it is higher, is, you know, it's intuitively reasonable to think it might, and I put a remark down here, because if they don't have to make those male parts, perhaps they've got more reproductive resources and they can make a few more ovules. And the way we can do this is we can use the fitnesses of these two types in the sort of phenotypic fitness model that I explained, although it's such a simple, genetically simple case that we don't need to. We can count the transmission to the next generation, and you can show that all you have to have for such a sterility factor to spread is that the females have to have a higher female fertility than the co-sexes, the original co-sexes. And that's because this is maternally transmitted. It's only the transmission through the maternal lineage, through female function, that matters. So it's extremely simple. And this trade-off between male and female fertility is an example of the principle of trade-offs and allocation of resources which are finite into either this or that we see very generally. It's very well known to exist as a concept. If you want to grow a large fruit, you have to take many fruits off the plant to make sure they don't compete for resources. You just leave one and you give it a lot of resources. They pour bottles of milk onto the soil around these pumpkins and you can get a very big fruit. These are real, it's a real phenomenon. And this is a central idea in the sort of things I'm going to be talking about. Now, these kind of simple situations that I talked about probably are not involved in this process, but I just want to go through a few interesting things about them, because you might think that that's the end of the story. A female can come to an equilibrium in the population because it can't go to fixation. But it's not quite that simple. Females can reach high frequencies in the population if they have a high female fertility. But of course, that will mean that the co-sexes are reduced in frequency, and that reduces female seed output. If the, well, it will tend to reduce female seed output. There's less pollen available from them. So the population would not necessarily become all female. If it did, it would go extinct. If the co-sexuals are unable to self-fertilize, then they also rely on the on the output of pollen from other co-sexuals, and it would be the same problem for them as it is for females, and then there would be nothing to stop females going right to a very high frequency and causing extinction of the population. But the usual outcome will not be like that, and in fact most of the species where we know this kind of thing are not self-incompatible, so that's all right. We would expect to come to a polymorphic situation. When you have a polymorphic situation, though, quite surprising result, at least at first sight, is that nuclear genes can restore the male fertility. A nuclear gene, a mutation at a nuclear locus that restores male fertility, even if you have the sterility genotype of your cytoplasmic genome, that can spread. So it's funny. Sterility mutations can spread, but then something which restores fertility can also spread. And the reason why it's instructive, the reason why that can happen, is because, as I just said, the sterility we've assumed was due to a maternally transmitted genome, cytoplasmic male sterility, and so any increase in female fertility is good enough to cause that to spread in a population. Whereas nuclear genes, to put it in a sort of very crude intuitive way, their interests are not just female fertility, they're both male and female fertility. And when you work out the population genetics, it turns out that they can evolve. So you can end up with a situation with two cytoplasmic genotypes, sterility and non-sterility, nuclear genotypes at a locus which is called R for restoration of male fertility. Let's just assume that we have two alleles and one is dominant. You might have a situation like this where only little r that have the sterility cytoplasm are actually female. If they have the sterility cytoplasm but they have a big R allele, then they go back to being hermaphrodite or cosexual. And of course, if they have the non-sterility cytoplasm, they're also hermaphrodite. And this kind of situation with both nuclear and cytoplasmic male sterility is known in quite a number of plants. In fact, the one I showed you, Silene vulgaris, where I showed you the pictures of the hermaphrodites and females, that's the genetics there. So nuclear restorer mutant alleles, mutations can gain a fitness advantage due to their male fertility, and they can invade even though male sterility could invade in the first place. The restorer can sometimes fix, and then you lose male sterility, or it can increase. You can have a polymorphic situation for both factors. It can become very complex, and it can, the genetics that I showed you in the previous slide is by far not the only possibility. You don't have to have these dominance relationships. You can have the situation where there are two different cytoplasmic types. They both cause male sterility depending on having the right genotype at nuclear loci. So you can have a specific restorer for this cytoplasmic sterility and a different restorer with a different genotype would be male sterile there. And so you can get all sorts of complications. But what I just want to show you this complicated slide to emphasize is that the restorer genes, the restorer mutations that arise can often fix. Male sterility could be lost. One cytoplasmic type could be fixed. It can lead to a difference in the cytoplasmic types between two species such that if you cross the two species, you would have male sterility in one reciprocal when it receives that cytoplasm, but not the restorers because the other species doesn't have the restorer genotype that it needs. So you can have the possibility of a constant kind of arms race situation where cytoplasmic male sterility arises, restorers arise, and you have frequencies of females on this axis and the left-hand panels here. Over generations, you get females appearing, and then they go away again. And this can happen repeatedly. These are just stochastic simulations of what you might get with different costs of the restorer. So I haven't mentioned any costs. I've just said male sterility cytoplasm might give you an increased female fertility, but you could imagine that the factors involved also come with some other fitness costs, fertility, survival. And in this case, the fertility is supposed to be affected by whether you have the restorer or not. And this is a small cost, this quite big, in fact, in terms of selection coefficients. This would be a larger cost, and this would be a very big cost. And what you see is with the very big cost, you can maintain these joint complicated polymorphisms. But if the cost is small, you're much less likely to. And that arises because you get these quite complicated situations. This is from a paper I published on the left a long time ago, which has the model, the genetic model I showed you a couple of slides back, with a non-sterility cytoplasm and a sterility cytoplasm. And I didn't label the axes here. One of them is the frequency of the sterility cytoplasm, and one of them is the frequency of the restorer, recessive restorer allele. And you can get all sorts of things going on. These are calculations of the recurrence relationships in infinite populations, and so you could come to an equilibrium ultimately given enough time. But you can see that in the course of these things, sometimes some of the frequencies get very, very close to fixation or loss. And so in finite populations, you wouldn't see full course of these things. You would, in fact, lose factors. and have to wait for new mutations. Just to show you that that's not the only genetic model, this is some results from another study where they assumed that they have two different cytoplasmic types, each of which can cause sterility if you don't have the right restorer genotype, and they can get other things. I gave the references here if anybody wants to look at these things, and they can get other extraordinary kinds of patterns of the evolution of these factors. And to this day, it's very difficult to do the population genetics of these systems. We can look at the edges of these parameter spaces and see there are places where you don't have any females present. There are places where you can have cytoplasmic inheritance with the restorer gene not present, and you can see when the cytoplasmic factor will invade and when it won't invade. Those kinds of things at the on the axes are easy to investigate, and here they've looked at the space in a similar way, but they've defined the regions where you can get different kinds of outcomes. But that's all that anybody is able to do, and I gave a reference on the reading list to a review of all this complication. But what I want to emphasize is that this is probably not the way dioecious species evolve as a rule, because you end up with these polymorphic situations, or else you lose the one of the polymorphic factors. You only go to nuclear systems by losing one of the factors. So that's all I wanted to say about cytoplasmic male sterility. If anybody wants to ask any questions now, I'm going to. There's one right at the back here. You have to speak very loudly, please.
Yeah, so I didn't understand why you were assuming or saying that hermaphrodite was the ancestral state for...
Sorry, I just can't hear you. Can you...
So I didn't understand why we are considering hermaphrodite to be the ancestral state for male and female.
Why this one?
Yeah, means why this is the case that...
So we're coming back to this one. Why I consider this to be the ancestral state? Well, I mean, observationally, we know that in plants, this is true. I showed you the phylogenetic tree. That could be replicated in other genera, and we know that there have been many transitions like that. If we want to talk about animals, we know that the most earliest branching lineages of animals did not have separate males and females. And well, sometimes we have cases where you do have separate males and females, but it's not genetically controlled. It's controlled by environmental influences. But you can surely think that the ancestor of any of these things had both sex functions. And that's what we want to explain. If you started with that, if we believe the ancestor was this, there's no problem to explain, but it can't always have been separate males and females. That's just simple common sense. So in this model, we've got two steps, at least two steps, as I mentioned. So we've had that earlier, a male sterility mutation and a female suppressing or female sterility mutation. And this has been modeled. So I'll look at first step one, using the phenotypic selection model. Yes? Well, we're looking at the evolution of separate sexes, so we're trying to find out how we can get there. Yes, lots of population, lots of plants, roses, yeah, apples, yeah, lots of plants. They have separate, they have male and female gametes, but in terms of gender, that's what I was emphasizing earlier, the plant is not a male or a female. It produces both sexes of gametes, so it's a cosexual or hermaphrodite. Some plants are hermaphrodites, as I mentioned in the first lecture, because they have separate male flowers and female flowers. Many pine trees have separate male cones and female cones, but the plant is cosexual. That's why the term cosex is so useful. One individual produces gametes of both sexes. I hope if I didn't make that clear enough, I hope it's a bit clearer now. So we can again work with this reproductive fitness, this concept of a hermaphrodite genotype, which we had already last time, by calculating the number of gametes that it will contribute to the next generation. And as we saw last time, that would depend on the fertility values, so how much pollen in the case of a plant, how many male and female gametes are produced, how much self-fertilization occur, how much inbreeding depression affects the progeny produced by self-fertilization. And we want to ask whether a male sterility mutant, a female mutant, will invade a population of co-sexuals. We're going to assume that we have a recessive male sterility mutation simply because that's the commonest type of male sterility mutation observed, and it's technically, of course, easy for the population genetics to work these things out when you have complete dominance, and we can use the phenotypic selection approach. We're going to assume, like I had in some of the models yesterday, that all ovules can be fertilized. We'll not worry about the possibility that pollen supply is limiting female fertility, although you could add that. We're going to have these symbols, selfing rate of the co-sexuals, some inbreeding depression, and again, female fertility, because it has some resources freed up, it could be higher than the co-sexuals, which have a more limited female fertility because some of their reproductive resources have to make their male parts. And when you do this, it turns out that you get this very simple equation, not as simple as in the cytoplasmic case. The increase in female fertility that females have relative to the co-sexuals has to exceed this amount. And I'm not going to work that out, but perhaps I should say that intuitively it makes sense because the co-sexuals lose progeny through self-fertilization and inbreeding depression. They put two of their gametes into each self-fertilized progeny, and those are discounted by the deaths or loss due to inbreeding depression. So their female fertility would be lower over than output. And that simple equation or simple inequality gives you conditions. For example, if there was no self-fertilization, then females would have to have double the fertility, female fertility of co-sexuals in order to be able to invade. This is a typo. Some inbreeding depression makes invasion much more likely, as you could see right away from the equation. And that's an interesting result because it says that you would expect some selfing and some inbreeding depression to make the invasion of females more likely than if you had a self-incompatible, completely outcrossing co-sexual species in the first place. So you would predict from that that self-incompatible hermaphrodites would not often evolve into species with this kind of polymorphism for females or into dioecious species, and that's what we see. Even if you look at plant families where they do have dioecious species and where self-incompatibility is common, they turn out to be, on the whole, in different genera. And so inbreeding avoidance, we can conclude, is probably one factor that's important in the evolution of dioecy, and I've already mentioned allocation of resources, and that's the other major factor. This is showing the invasion by females, plotting what that equation looks like as on the x-axis as a function of the selfing rate. That's the equation again. And you can see that if you have some selfing and you have some inbreeding depression going on affecting the progeny of self-fertilization, it gets easier for females to invade the co-sexual population. So that's obvious from the equation, and it makes sense, and it fits the empirical result. The only thing that was puzzling when we first did this piece of analysis was that there was a belief that organisms were either outcrossers that suffered from inbreeding depression, or they were inbreeders that didn't suffer from inbreeding depression. There was a very crude, non-quantitative view of mating systems, and I hope I gave you a slightly more quantitative view that has been much more widespread since people were able to estimate mating systems using allozymes. In my previous lecture, I showed you that often selfing rates are intermediate, and inbreeding depression certainly can be detected in organisms that are not fully outcrossing. It's less in organisms that are highly inbreeding, but it exists wherever you look. So you probably have to have some self-fertilization, some inbreeding depression, and if you didn't have any increase in female fertility of the females, the inbreeding depression would have to be very severe, probably more severe than, given the selfing rate, than seems plausible. Probably both factors are likely to be involved. We can see that. I'll come back later to this part of the diagram, the invasion by males. So I've already mentioned allocation of resources is very important, and I want to emphasize this because, again, people took this theory often to say, oh, the conclusion is that separate sexes evolve to avoid inbreeding. They do have that consequence, and it is important in that equation. The inbreeding depression comes in as an advantage of being a female, but it's not the whole story. The allocation of resources and the fact that females might be able to have higher female fertility than the co-sexuals is also important. Now, we can test this theory because, as you saw, it was quite quantitative in terms of the equation, and the items in the equation are things that we can actually measure in real organisms. We expect a polymorphism, as you agreed here, of females and hermaphrodites if we have a nuclear factor like this, and we do know that there are gynodioecious species. Unfortunately, many of them are not suitable for testing this nuclear gene theory because they are gynodioecious species with females and hermaphrodites due to cytoplasmic male sterility. So we have to do genetics and determine that a species we'd like to test the theory on is really segregating for a nuclear male sterility factor. And there aren't many, but there are some. This is one work of Josh Cohn at the University of San Diego, though he wasn't there at that time. It's a cucumber species that smells terrible. That's why it got this name. And he did this heroic study of this organism. It lives in the desert, and an individual can be several hundred meters long. It's a cucumber species that grows long stems. But he did self-pollination and cross-pollination in order to estimate the inbreeding depression. That was our delta in the equation. And he detected inbreeding depression at various stages of the life cycle. Seeds of self-pollinated plants of the co-sex are much less able to, they produce less seeds than if you do a cross-pollination. The seeds seem to survive equally well, more or less, but the young plants survived worse if they were, that's cross-pollinated, yes, that's the survival, good for cross-pollinated, bad for inbred, and so it went on. So he was able to estimate that delta, taking fairly reasonable amounts of the life cycle into account, and the delta value he got is indicated on this diagram. So I've got inbreeding depression now plotted on the x-axis, and the predicted female frequency from the equation. That's the inbreeding depression that he estimated. He didn't have a very good estimate of the self-fertilization rate. It wasn't easy to get that, but he estimates it to be about this level. And I just plotted 3 different self-fertilization rates of the co-sexuals and what the equation predicts. And this is what the inbreeding depression that the selfing rate that he estimated gives us. And this is the frequency of females in the population, and you see it's spot on where we have the estimated selfing rate and the estimated inbreeding depression. So it's probably slightly coincidental that the agreement is so good. I didn't choose this one because it's so good. I chose it because it's one of the few cases where the genetics probably is a good nuclear male sterility factor. Okay, so is everybody happy that we can reach this stage in this diagram and that it involves inbreeding avoidance and females getting a female fertility advantage? Any questions there? Pretty straightforward. How can males evolve? You can see that we should consider males first coming from a co-sexual. We can consider this kind of mutation. We can do the same population genetics analysis now for a female sterility mutation. And we're going to do that as a problem, so I won't do it now. But what it turns out is that males can very rarely invade populations. It's much more difficult for that kind of mutation than it is for this kind of mutation. The conditions are much more stringent. And we'll discuss, if we have time in the problem class, where that asymmetry comes from. But it's simple to see it. So that's pretty strange. We can get co-sexes to produce females. We can't get co-sexes to produce males and have them spread in populations. And empirically, this asymmetry corresponds with an asymmetry that's real in the real world and was noticed even by Darwin when he studied plant mating systems and looked at what is known in real plants. And as we saw, male sterility polymorphisms, gynodioecy, that's found 5% or so frequency in different floras. Androdioecy, males polymorphic with hermaphrodites, Darwin couldn't find a single example. Examples are now known, and if anybody wants to ask me about them, I can explain about them. But the bottom line is that these androdioecious populations that have been discovered over the time since Darwin either turn out to be fakes, they're not really androdioecious, they're kidding their pollinators, or they are cases of breakdown of dioecy where you have a reversion such that females, like in this arrow, have reverted back to co-sexuals and they coexist with males, which is a different situation. So it's another case. I already mentioned evolutionary reversals can happen. And sometimes, although it seems counterintuitive that you could evolve down to here and then it would be advantageous to go back, change in the ecological situation, of course, can make being co-sexual much more advantageous than it was for this population. So if pollination fails for some reason, you really don't want to be female. It would be better to mutate, get a bit, have a back mutation, and get a bit of co-sexual function again. Okay, so I'm not going to talk about that side at all because it's theoretically predicted to be rare and empirically appears to be rare. So what about this situation, though? Because now I've told you males can't invade, but now I'm going to tell you that that's ignoring something important. Males have less problem invading once females are there. Here we looked at co-sexes and asked whether males can invade. Here we're looking at a population that's got females in it. And now you intuitively should see that, of course, it's more advantageous for males than before. The hermaphrodites might easily undergo a mutation. It might easily be advantageous for a mutation that makes them allocate more resources to male function than when females were absent, because now there are opportunities for that, for those male gametes, to fertilize the eggs, ovules of females. So hermaphrodites now gain fitness through fertilizing females, whereas before they would have to have a big increase, implausibly big increase in male fertility to do so. It turns out when you do the population genetics of it, that male fertility, in this case once females are present, must increase more than female fertility. The co-sex's female fertility is decreased. So you've got to have reallocation of resources such that you get a big boost in male fertility and not too bad a decrease in female fertility. And this shows you conclusively that you can't evolve the second step without reallocating some resources. So again, I'm really emphasizing that it's not true, that it's just an inbreeding avoidance mechanism. When we get the equation for the increase of males when females are already present, this shows some of these results. So this is the level of inbreeding depression. This is the predicted frequency. This thick line shows the predicted frequency of females at equilibrium that you can work out from the population genetics. This situation, and we're asking if we had a rare male, can it invade? And you can see that it can invade after inbreeding depression is higher than this level, and females start to become common. Then, according to how much male, this shows the this is showing, for a particular case, is showing threshold recombination rates. No, I should have drawn another diagram then. Sorry, I got myself a bit lost. I need another diagram, or an addition to this diagram to show this. Okay, so we're going to have a male sterility mutation, and now I'm going to write in some of the genetics here. So we've got a chromosome, and we're going to have a recessive male sterility mutation. These are going to be females. And they're going to be polymorphic with the co-sexuals, which are going to be at that locus, have the dominant allele, like that, or they could be heterozygous. In fact, so you could have both. All right, that's the genetics I said we were assuming. And then you could work out the population genetics for the males to invade. And what you can see intuitively, again, to start with the intuition, is that if you have a male sterility mutation, like in the females, it would be very harmful to have a female sterility mutation. So that we're now looking at a second locus mutating, thinking about this step. If you have a mutation at very close to this locus, for example, it would turn the hermaphrodites potentially into males, or make them more male-like. And if it was very closely linked, it could be advantageous because of the advantage I just explained. But if it was very far away, it could be advantageous in the individual it occurred in, but recombination crossover events will occur between one generation and the next, and individuals with the little m recessive allele will get that female suppressor, and that will be disadvantageous to them, and it will make it more difficult. And so it turns out that there's a threshold recombination rate, and that's what this is showing on the y-axis, for the invasion of that second sterility mutation. So that's an interesting result because it suggests that we should have the second step. I have to draw the genetics again here. The females will be at the first locus, little m, and at the second locus, now we can say, we assume that they have a female fertility factor or something like that, and the males will not have little m, and they have a dominant female suppressor at a second locus. And in fact, that locus needs to be in the genome somewhere close to this first locus. It's not occurring on the same chromosome, in the same haplotype as the male sterility, otherwise you would get the bad effect, but it has to occur at a locus which is in a genome region linked reasonably closely. Okay? So this is, to put it in more population genetics terms, there's a condition for the invasion of that second gene that's not just is it advantageous or not, the recombination between the two loci comes in, and it makes a more complex condition. And that's a sort of situation that you can have in other population genetics models, other settings, where you have an interaction between two genes. This is an example, a specific biological case of the phenomenon of epistasis that Brian talked about, where the effect of a mutation is different on that background from this background. It's good here, it's bad there. And in those cases, it was already known in population geneticists working out models just in general, thinking about epistasis, that you could have situations where a mutation at the second locus would not invade if it was too loosely linked, even though it could invade if it occurred at a linked locus. So in this case, we're not just writing down fitnesses, we're allowing the fitnesses to develop from a biological model of what the mutations are doing. We're change to be male sterile, for example, and that allows us to calculate an expression for the reproductive fitness. And we're working out the consequences of those fitnesses that arise from a biological model. And we're not just writing down, like Brian did, a matrix of possible epistasis that gives possible epistasis. The epistasis is also arising from the biological situation. So it's using the results are based on the same sort of population genetics, but the fitnesses are arising in a specific biological situation, just to make it more concrete, really, for people who have those kind of preferences. Okay, so I already talked that was out of order, so I'll just skip on, I think. Now, what we know is that you might wonder, well, why did I assume a male sterility mutation? It could be a gradual process. What we think is that a male sterility mutation will not necessarily be advantageous unless it has a pretty big effect on male fertility. If male fertility is reduced a little bit, it's not very likely to affect the self-fertilization rate. If you produce half the amount of pollen the co-sex produces, it might still lead to about the same amount of self-fertilization. So to the extent that inbreeding avoidance is involved in this evolutionary pathway, the sterility, rather than just a reduction in fertility, is likely to be involved. But that's not true for the second step, where we're thinking about the co-sexes evolving into males. They could become a little bit more male, because the main advantage they are getting is not through mating avoidance, it's through being better at fertilizing the female gametes of female. And so a gradual adjustment might well be important. So where I drew a single arrow, it might be multiple evolutionary steps. And again, this is supported empirically because we see plenty of examples. It's extremely common for dioecious plants to have females that are female in any environmental circumstances and very strictly female, never produce any male, have any male function, whereas males often include co-sexual forms, and sometimes in certain environmental conditions they can produce fruits quite a bit, and in other environmental conditions not. So this is just showing proportion of flowers with fruits. Females always, so this must be not pollen limited, to practice that term again, whereas the individuals that do produce pollen sometimes produce no fruit in their flowers, but sometimes produce fruit quite a lot. So that's one of our assumptions of the model in a way, or it seems reasonable to think in these kind of models that it might be a gradual process. So it's not just necessarily a two-step, two-mutation process. Multiple mutations might well be involved. The other assumption I made was the trade-off that you could have males that are not fully males, and that being a better male might entail being a worse female, and that seems to be supported by some recent data that have been published in Silene latifolia using different kinds of mutants, and it seems as if there's an inverse relationship. A plant, this is a wild-type female and wild-type male, but plants can produce flowers in between, and if they've got mostly male function but not full wild-type male function, they seem to have a bit smaller female parts, and if they are in between, they're in between for both, and if they're nearly female, they have very small male parts. So it's reasonable to think that these assumptions may hold in real organisms. Okay, so we've got here sex chromosomes is another question. It's a separate question. Why do we have sex chromosomes? I haven't talked about that at all. I've just said now we've got separate sexes. But this result is very illuminating about that second question, because now we have a situation where we've got, in terms of what Brian was talking about, we've got two different haplotypes. Females are homozygotes for this haplotype. Males will have this haplotype with two differences at two loci, or more than two loci. And we know, again, from basic population genetics, that that kind of situation with an interaction will produce a situation where selection will promote less recombination between those loci. The males will be most likely double heterozygote, and recombinants will be disadvantageous. Recombinants could carry the male sterility factor and be female, but they can carry the female suppressor as well, and that's not good. So you expect a region that has undergone this kind of evolutionary event, that it might evolve suppressed recombination in this region to keep the right combinations of alleles together. So if you establish a two-locus polymorphic equilibrium, it selects for reduced recombination. And this can explain why sex-determining regions become non-recombining. So that's a very quick, simple. It falls straight out of the concept that we have at least two mutational steps that have to spread and become polymorphic in the population. Okay? But it doesn't answer the question why, if you remember back to the plant sex chromosome diagram I gave, and I said it's much the same in humans and even more extreme in Drosophila, why do the whole chromosome pair, or nearly all of it except a bit at one end maybe in humans and the plant, but not even that in Drosophila, why do we have large non-recombining regions? Why don't you just suppress recombination, say move a hotspot, evolve a hotspot somewhere else and take it away here? That doesn't seem to happen. So this is the process we've got, we've come to so far. It's just the same diagram where we've had step 1 happen, we've established co-sex and female, we've had step 2 or maybe 2, 3 and several other steps which have established males. I've called it a suppressor of femaleness here. Instead of just big F, I've called it this, but it means the same. So this we could call a proto-Y chromosome. There's no recombination suppression. It's just got two linked factors. Why do we end up suppressing recombination across the whole chromosome, or nearly the whole chromosome? That's the question. And the answer that people think is most plausible is that now we have males and females have selection acting on them in a different way. Male benefit mutations can occur that would be bad for females, just like we already saw, or you could have female benefit alleles. So we're going to evolve the sex-determining region, say, might not recombine. It's for the reason I just said, it's somehow managed to evolve suppressed recombination. The X chromosome is still a normal recombining region, of course. And then if we had an advantageous mutation for males, this other allele would be the one that's better for females. And if you have the male benefit allele be bad for females, then you could establish exactly the same kind of situation. Again, it's not sterility now, it's other factors that interact with sex, with the sex-determining region that determines gender. And you could establish linkage disequilibrium with the male benefit allele mostly being on chromosomes that carry this male-specific region, and the female benefit allele mostly on the other chromosomes. And if you had recombination, that would not be so good, just as I already explained. So this region that still recombines is called the pseudo-autosomal region, PAR, pseudo-autosomal region, and recombination between sexually antagonistic factors that have established polymorphisms in that region. There will be another two-locus polymorphism where the whole Y chromosome fully linked region is one allele, essentially, doesn't recombine. And this is now like a two-locus polymorphism, just as before. It would select for recombination to become suppressed in this, across this new region. And that might explain the whole thing. So suppressed recombination, it occurs in one initial step, and then it might select for a new region to suppress recombination. That's the hypothesis. It's not yet confirmed by empirical evidence very far, but you can see that it makes sense. But we do know that the recombination wasn't suppressed in a single step. We know this in several different organisms. If you take humans, for example, this is a a picture of the X chromosome in humans, and this x-axis here is the genetic map of various genes that have been mapped on the X chromosome that still have alleles, copies on the Y chromosome. Most genes, as you know, as I mentioned earlier, that are on the X chromosome have gone from the Y chromosome, but some genes, a few genes, a handful, are still present. And you can use the divergence on the y-axis, the sequence divergence of sequences of X chromosome alleles from their Y chromosome counterparts to get an estimate of how long recombination stops. So Brian already mentioned that you can have subdivision within a species, as it were, genetic subdivision. And here's the case of genetic subdivision. The X chromosome is a population that occurs in females, and in males the Y chromosome occurs only in males, the non-recombining part, and it's as if it was an isolated population. It doesn't recombine with the X, just as if it was isolated geographically in another distant population and never recombined. So you can have sequence divergence just as if it was in a separate species even. So there's a certain sense in which it's true to say that males and females are different species, but only in this limited part of the genome. And you can see that genes which are mapped at one end of the X chromosome have high divergence, and as you go closer to the recombining part, there seems to be a gradient with smaller divergence. And exactly the same thing is seen in this plant Silene latifolia. These are data from the postdoc in my lab got these data. We have a genetic map where these much smaller number of genes are mapped. These genes all have copies on the Y chromosome. It's not looking at any genes that have got lost from the Y chromosome. And we can estimate XY divergence for silent sites, which roughly estimates time. You can see some of them seem to stop recombining longer ago than these. So even this longer ago time, the divergence is quite small. It's like this divergence in the human one. And here's another plant, papaya. Again, we have some region where we think divergence is quite high, although you can see standard errors are very high, and some other region where XY divergence is smaller. And again, it goes with the map of the X chromosome, as if one part stopped recombining first, and then after some time, much more time, another part stopped recombining. So we know it didn't all stop in the sex-determining region in one event. And so we would end up with the X and the Y looking very different from one another and not recombining, they only pairing at the tip. Now, the reason they look very different from one another, there are various reasons, but one major reason is just loss of genes from the Y chromosome. So I'm going to talk a bit about that. In the human Y, as I already told you, there are very few Y genes. Some of them are repeated several times, but if you take different Y chromosome genes, compared with, there are 24 here, compared with 1,000 or so in the X. And similar in Drosophila, except that there's no known gene on the Drosophila Y chromosome has a copy on the X at all. It came from somewhere else. Now, Brian's already talked about the consequences of suppressed recombination, so I'll go very quickly through it. But the Y chromosome is, as Brian said, an asexual part of the genome, doesn't recombine. I'm talking about the non-recombining part. This evolves, and if you have a selective sweep, it's going to go through the population. It's, oh dear, wrong bit. Is that the right one? An advantageous mutation sweep through the population, and this haplotype over the whole non-recombining region will be fixed. So we will lose diversity, and we do indeed see low diversity of genes on Y chromosomes. If such an event happened and there happened to be a deleterious mutation on that chromosome, that gets fixed too, just as Brian explained. So that's going to make Y chromosomes fix deleterious mutations that would not have been fixed if recombination occurred. We can also, as Brian explained, have removal of deleterious mutations, again, X's, leaving us with less variants than we had before, and that will occur in a non-recombining region, as Brian explained. I'm not going to explain Muller's-Ratchet, because that's another process that's somewhat similar to the one I just showed you, and I don't have time, but some of you may have heard of it, and if not, it's described in our textbook. It's probably an important process happening on Y chromosomes and asexual genomes. So what we expect to see is that we have an early stage where you've just got the two loci, or maybe a few loci, sex-determining loci, that recombine, that don't recombine on the proto-Y, and that expands through these processes of sexually antagonistic mutations, perhaps leading to this situation that will select for suppression of recombination. So we end up with the Y looking peculiar, not like a normal chromosome. And then we can expect that we have loss of functional genes or loss of function of genes, so that perhaps they can be deleted and lost, and we would end up with only a few genes that are on the X remaining on the Y. That's the idea. So we expect the Y chromosome genes to undergo more deleterious substitutions. We expect insertions of transposable elements to be able to go to fixation on the Y chromosome when they would not, and even ultimately, once genes have lost function, or even just maybe their function is not so good as the X, you could do without them entirely, and deletions can occur on the Y, so the Y could end up very small. So in the plants, we think it's reached the stage where these first two processes have happened, and the insertion of transposable elements explains why the Y chromosomes of plants are often very big compared with the X. Lots of insertions have occurred. And then in later stages, like in humans, the parts of the Y may become deleted, and that's why they have small Y chromosomes compared with the X. So we've done a bit of work, and I'll have a few minutes to talk about it, in Silene latifolia, this plant I've mentioned. This was done by this postdoc I mentioned, Roberta. She could get RNA sequences and use those in family, parents and offspring, to see whether you've got any sequence variants that show sex-linked patterns of inheritance. And she was able to find sex-linked patterns of inheritance, SNPs, as you've heard about before, that identify a piece of sequence as a sex-linked gene. That's cutting a very long story short. Because these are RNA sequences done by high-throughput sequencing, we have very crude, but we have some estimates of their expression level. So this graph shows you the numbers of reads from the X chromosome allele and Y chromosome allele for a whole bunch of genes which we're fairly sure really are located on the X or Y chromosome. And you can see there's a strong tendency for lower expression of the Y-linked alleles. These are the same gene in the X and the Y of males. It's just a bar chart showing you the same thing. There are some genes where the Y allele is expressed at a higher level than the X, and of course some of this is just experimental noise. We're estimating expression crudely, but there's a predominance of low expression. Now, low expression on the Y might be advantageous. It might be that these alleles would be advantageous to have lower expression in males than females, but some of them probably would be deleterious changes. We can also look at deleterious changes using this kind of experimental design. So I'll just mention it because we haven't really heard yet. We've heard a lot about, a certain amount about phylogenetic trees, and one thing that wasn't mentioned is that when we're doing these kind of analyses, we want to have a phylogenetic tree and know the relationship of species of interest to their relatives in order to have an outgroup for this kind of comparison. So in this case, this other species I showed you the picture of, Silene vulgaris, is our outgroup species. We believe that it has never had an ancestor with sex chromosomes. It's never had sex chromosomes in its ancestry. And here's the species we're interested in that has an X and a Y sequence. We could look at a particular locus. And we can compare the sequences in these three cases, and we can use the comparison to infer the changes that occurred in the X lineage and the changes that occurred in the Y lineage in relation to what was present in the other two. So X, if X is out, the odd man out, and the Y and the outgroup species agree, we guess that's a change in the X lineage. If the Y is the odd man out and the X and the ancestor agree, we guess the change has occurred in the Y lineage. And when you look at this quantity that Brian was mentioning, divergence for non-synonymous sites divided, just to take care of mutation rate differences, divided by synonymous site divergence, we see a lot of high values over on the right-hand side of this graph here for the Y lineage, should be. High values for the Y lineage and compared with the X lineage. We're getting a lot in that lineage. We're getting an unexpectedly large number of non-synonymous changes controlling for the mutation rate. So it's not just a high mutation rate, though I drew this line longer because there is a higher mutation rate as well, but we don't need to worry about that at the moment. So we do have evidence that, I call it degradation of sequences. The Y-linked copies of genes which are on the X and the Y-linked ones are strange. They're odd man out relative to the outgroup and the X. Now, I've talked a little bit about homologous genes on the X and Y. I've mentioned genes lost from the Y, and I've mentioned the pseudo-autosomal region genes. And, of course, these genes may, the ones that are still present, may also adapt and evolve. You know, there may be mutations which are good for males, bad for females. And that kind of thing has been very much studied as well. You can ask this in several different contexts. You can ask if you've got a copy on the X and the Y, how does the X and Y copies evolve differently? The one on the Y is confined to males, the X one appears in males and in females. If a gene is lost from the Y, its evolution is also going to be affected because now it's present in males and females, but it's present in females twice as much as it's present in males because females have two X's and one Y. And then we have these regions, the pseudo-autosomal regions. I'm just showing the same sort of diagram again, where you have a part at the end of the chromosome that does recombine, and whereas most of the chromosome doesn't. And as Brian mentioned earlier, if you have balancing selection, which of course the sex-determining locus is a case of a locus under balancing selection. We have maintained both X and Y copies because we have usually about a 50-50 sex ratio and everybody has a male and a female parent. There's balancing selection. It may not be a familiar way to think of balancing selection, but it's a very familiar example that is in fact a case of balancing selection. That's going on. And as Brian mentioned, regions linked to a locus under long-term balancing selection show linkage disequilibrium and signals of association with that. So you have all sorts of things going on, and I don't have much time to talk about those things, but I'll just say we know very little about loss of genes from plant sex chromosomes. Loss of genes has been studied extensively, just described extensively in humans, for example, and Drosophila. And those are interesting findings. We want to explain them, but we would like to know much more about the plant cases simply because, as I've shown you, plants evolve separate sexes and sex chromosomes when they do evolve sex chromosomes. That's happened quite recently. So we want to know what's happening early. We do know that a lot of genes in plants are expressed in the haploid stage, the pollen and ovules, and so perhaps they can't be lost, but we're starting to investigate that. If you had genome sequences, you could just look at the X. So here's a real case. This is papaya. It's only got a small non-recombining region, so it is possible to sequence that region. And we could look at the X, and we could write down a list of all the genes that are present, and we could ask which ones of them are present on the Y. So in this particular example, there are 47 genes that I've drawn here that are present on both the X and Y, and we could catalog ones which are missing from the Y, but they're still present on the X. It turns out that we don't see a dramatic loss from the Y, and it's not at all clear, in fact, what's happening in papaya. So this is just to show you the ideal. That's what we'd like to have. We can't yet do that in any other plant. But if we did have genes that are lost from the Y, or even genes which are not very well expressed, not very functional on the Y, you might expect that males would evolve compensation for that because they have an X with a good set of genes and a Y with a not so good set of genes, and perhaps they would show increased expression of the X-linked genes so as to compensate for that, get back to the optimal, proper level, if you just vaguely imagine that. And we do know that this dosage compensation phenomenon has evolved in Drosophila. It was first discovered, I believe, in Drosophila a very long time ago. If you look at the genes in our own study in relation to the level of expression, the most interesting ones to look at would be ones where the expression of the Y-linked copy is pretty poor. Most interesting ones would be to look at the ones where the Y-linked, where there isn't a Y-linked copy, but that's not so easy, as I said. If you look at ones where they have low Y expression of the Y-linked copy, the way I've drawn this here is that I've divided the level of expression in females into two bars to show two X chromosomes, and then I've compared the X in black and the Y in less black, in grey, and you can see there's not much difference. So there's dispute at the moment in this particular plant. One analysis claimed that there was dosage compensation, and using exactly the same approach, in fact, we'd already done this, but I think we have a more reliable set of sex-linked genes. We don't find it, and no doubt it will be sorted out at some point. I've talked about sexually antagonistic polymorphisms, and I don't have much time to talk about them more, but I will just mention that the population genetics of these kind of cases can be done as well, this kind of gene, and we can show that the pseudo-autosomal region is more likely to have this kind of polymorphism. I said that if in the recombining region you establish a polymorphism, so that you have a two-locus polymorphism for a sexually antagonistic gene and a sex-determining region, that could select to suppress recombination in a new part of that chromosome pair. And you can show that if you have sexually antagonistic mutations, which is not entirely clear, they might be a very, very rare kind of mutation, that you would be able to establish polymorphisms. This shows for a particular selection coefficient in males and females. It shows that if you have unlinked a locus here with these two, the region where you have polymorphism is just between those two lines, you may be able to see. But if you have tighter linkage, so pseudo-autosomal genes would be lower recombination than 0.5, there's a wider region where polymorphism can be maintained, just as you might expect. And when you maintain such a polymorphism, of course, it's really a case of linkage disequilibrium, an association with the male determining region, the male benefit allele gets associated, and you would have allele frequency differences. If you took a sample of Y chromosomes and X chromosomes, they would have allele frequency differences. And this picture on the right just shows, for different recombination rates, what in theory, in infinite population theory, you would get in the way of allele frequency differences. With strong selection, one over here, you can have considerable allele frequency differences, and with weak selection, obviously, it's less likely unless you go to very tight linkage. So I think I should probably stop at that point. I had another, because Kavita asked us to talk about our own research. I had another bit of it, but I don't think there's really time for it. I think it'd be better if people ask their questions now in the last 5 minutes. If there are any questions? Yeah, one other. Yeah, well, I did touch on that yesterday in the sense that I talked about the ecological factors, that, of course, this is the evolution of gender, not sex. Having separate sexes definitely is a risky phenomenon for females, or for both sexes. They may not be near enough to a mate, or the pollinators may not function well enough to give them enough mates. So it's a good question because, as I said, the sensible thing would be a hermaphrodite. But you can see the evolutionary factors, avoidance of inbreeding and reallocation of resources, that can lead you to evolve separate sexes. But in the long term, the environment might change so that now female or male fertility or both are not good. And then, as I said, it would be advantageous for either females or males to have a back mutation so they are able to function at least enough as the other sex to fertilize their own gametes and produce progeny on their own. So it's, as I said, it's an example of reversal of evolution, could well happen in these situations. And indeed, we see that. If you look at the phylogenetic tree of groups of plants where separate sexes have evolved, you very frequently seem to see reversals back to hermaphroditism. It's interesting that it's a fairly, separate sex is a fairly rare condition in plants. That's why we work on plants when we were interested in its evolution, because it's rare and it's at the tips of phylogenetic trees. We usually think of that as indicating a disadvantageous state. Asexuals, for example, they are found, if you do phylogenies, at the tips of phylogenetic trees, as if asexuality can evolve, which makes sense from the big advantage, transmission advantage that it has, but it doesn't last very long because of the deleterious consequences that follow on from having become asexual that Brian explained. So we normally think that makes sense. Asexuals pop up often and they fizzle out quickly, and so we see them only at the tips of trees. Having separate sexes has that same pattern for many, many groups of organisms, though not obviously for many animals. And so, but it's not a disadvantageous character. It has to have evolved. It has to evolve through a series of evolutionary changes that were each advantageous at the time. But again, if the ecological circumstances change, it could end up being bad. So you have to think about both time scales, and it's sort of a bit surprising to people at first, I think, that things can evolve, but down the line will prove to be disadvantageous, especially, of course, if circumstances change. It's no surprise that the advantages are no longer applicable. That's a good question, though. Any other points anybody wants to raise? Yeah, Kavita. Yes, it's the commonest. Most animals, it depends which kind of animals. Most familiar animals, mammals, insects, are not. But a lot of fish, a lot of mollusks are hermaphrodites. We just sort of forget that we were told quite rightly not to call them the lower animals, but you know what I mean. There are lots and lots of hermaphrodite animals, and more hermaphrodite animals than dioecious animals, if you want to count by species, so I'm told. I haven't done it, but I believe. We just think of the animals with 4 legs and so on, and of course they're not hermaphrodites. 4 or 6 legs, I should have said, or 8. Okay, I believe it's lunchtime. Is there no more questions?